Hi class, let's start our discussion on prokaryotic metabolism. This discussion is going to be broken down into several sections. We'll talk about enzymes, we'll talk about metabolic pathways, and then we'll end with a discussion on specific metabolic pathways, particularly the ones used to generate ATP, and then some alternative metabolic pathways. This traditionally is kind of a meaty chapter for most students. So let's put on our thinking hats and get started. First, let's talk about an enzyme. We talked briefly about enzymes earlier when we were talking about our four main macronutrients: proteins, lipids, carbs, and nucleic acids. Enzymes are a protein that acts like a catalyst. All of our metabolism, all of the chemical reactions in our cells need to be regulated, and are going to be regulated by these enzymes. Metabolism refers to any chemical reaction that is inside of the cell. These chemical reactions can be broken down into two big categories. That can be an anabolic chemical reaction, or that could be a catabolic chemical reaction. An anabolic chemical reaction takes smaller molecules, connects them to make bigger molecules. So if we're building a big structure, molecularly speaking, in the cell, generally speaking, this is going to require the input of energy. Just like when you're building a house or you're making something out of Legos, it's always harder to create than it is to destroy. We also have catabolic chemical reactions. With catabolic chemical reactions, we start with a large molecule, and we break that larger molecule into smaller molecules. This process of tearing down and breaking apart large molecules into small molecules releases energy. A catabolic pathway, generally speaking, is going to be easier to go through than an anabolic pathway. So, here we can start off with a big complex 6-carbon glucose molecule, and we can break that glucose molecule down into a 3-carbon pyruvate molecule. So as we go from a bigger molecule to a smaller molecule, we're losing energy from the molecules, and that energy is going to be yielded out to the cell to be harnessed. This is going to be a catabolic process, and the molecules are going to be less complex. We can then take these small precursor molecules that we have, there's the mouse again, and we can start connecting them to each other to make more complicated molecules. This is building up to make more complicated monomers. We can then take those simple monomers, connect them to each other to make macromolecules. Then we can take those macromolecules and connect them to each other to make an entire cell. These processes are taking smaller molecules and connecting them to make larger structures. So they're going to be anabolic pathways that have a net input of energy. As I mentioned before, I got ahead of myself a couple slides ago. We need some kind of a catalyst, some enzymes to speed up and regulate these anabolic and catabolic chemical reactions. It's worth emphasizing that an enzyme or a biological catalyst is not used up during the chemical reaction, so it's never going to become part of the product. These enzymes can be recycled. They can be used over and over and over again. These biological catalysts are going to speed up the rate of the chemical reaction. Now, if the catalyst was not present, the reaction would still take place, but it would take place so incredibly slowly that the cell would not be able to live. We need these catalysts for the reaction to take place in a timely fashion. So here's a wonderful figure from your textbook that's great fodder for making a flashcard about enzymes. Enzymes are made of proteins. Some of these enzymes are going to require cofactors or coenzymes. Think of those as vitamins and minerals. Our enzymes are an organic catalyst. A catalyst is just something that speeds up the rate of chemical reaction. Our enzymes are going to be very unique. The unique shape or structure of our enzymes dictates what they can or cannot do. It's going to dictate the specific molecules they can interact with. Our enzymes control metabolic pathways by speeding them up to the point that we can remain alive, or the cell can remain alive. On the enzyme, we have active sites where the substrate molecules, the target molecules, will attach to the enzyme. Our enzymes, generally speaking, are going to be much larger than the substrates or the molecules that they interact with. Our enzymes can be very loosely associated with very specific reaction products or integrated into reaction products, but they aren't necessarily going to be permanently integrated into the reaction products. Remember, enzymes are not going to be used up during the chemical reaction. Our enzymes can be used over and over again. Since we don't use them up during the chemical reactions, we can recycle them, so to speak. Our enzymes can be very affected by temperature and pH. This is one of the reasons why if you heat up or cook your food, you can denature that proteins or the enzymes that regulate the chemical reactions that keep bacteria alive. This is one way that cooking food sterilizes food. And then finally, our enzymes can be regulated through feedback loops where the end byproducts or the beginning genetics that control the enzymes can be controlled by the products made by the enzyme's metabolic pathway. So let's talk about how these enzymes work. Our enzymes are going to have a physical site where the substrate, and the substrate is the molecule that the enzyme was going to change in the chemical reaction, can bind to. Since the enzyme has a spot for that substrate molecule attached to it and be wrapped around, the enzymes, generally speaking, need to be larger than the substrate. So here we can see a large enzyme, and this large enzyme is going to typically have an indentation in it. Those indentations in the enzymes, generally speaking, are referred to as the active sites. And the substrates would go into those active sites, or those indentations in the larger enzyme molecule. Once the substrate is bound into that active site, there is going to be some kind of a chemical reaction that occurs in that substrate while it's in the active site. There'll be a change to the substrate. The enzyme, as I mentioned before, and I'll say it again, is not used up by the chemical reaction. It can be used over and over again. If we look at our enzymes, these are generic apozymes. An apozyme is an enzyme that has not been fully activated yet. An apozyme is an enzyme that needs to be tweaked just a little bit to be activated. Here we can see a metallic cofactor, or also known as a mineral. Sometimes we need minerals in our bodies to fully activate our enzymes. Here we can see a coenzyme. A coenzyme is also known as a vitamin. So some of our enzymes will need a vitamin to be fully activated. This is one of the reasons why it's kind of nice to take a multivitamin pill, or to at least eat a wide variety of food and get a balanced diet so you can get your vitamins or minerals, aka metallic cofactor, also known as your coenzymes and your metallic cofactors. You want your apozymes within your metabolic pathways to work properly. Sometimes the enzymes require both the coenzyme, both the vitamin, and both the mineral, both the metallic cofactors to work. So sometimes we'll have an enzyme, though, that doesn't need to have a vitamin or mineral. Those are just going to be a simple enzyme. They consist of a protein all by themselves and can work all by themselves. Sometimes, though, if our enzyme needs to have that cofactor or that mineral attached to it, that cofactor or the coenzyme attached to it, we will call that the apozyme, or sometimes also referred to as the holozyme. The holozyme is going to be the conjugated portion. It's going to be the inactivated enzyme plus the vitamin combined. The apozyme is just the inactivated enzyme, and then the cofactor or coenzyme is going to be that vitamin or mineral. When the substrate binds to the active site within the enzyme, so if we go back a couple slides here, when the substrate molecule is stuck inside of that substrate, so it's stuck in the groove in the enzyme, we'll have a temporary enzyme-substrate complex form. Now the substrate, the shape of the substrate is so specific to the shape of the active site that you could think of the relationship between the enzyme and the substrate as kind of like a lock and a key. Only the correct shaped substrates will fit into the correctly shaped active sites on the correct enzymes. This is important because when we are regulating the chemical reactions that keep us alive, we want the correct enzymes regulating the correct chemicals for the correct metabolic pathways. Once we form this bond between the substrate and the enzyme, we're going to initiate the chemical reaction within the substrate. Now these bonds that form between the enzyme and the substrate, generally speaking, are pretty weak chemical bonds. They're going to be hydrogen bonds, and they're going to be easily reversible. This enzyme-substrate complex is formed and can typically require the aid of a cofactor or a coenzyme, particularly if we're working with a holozyme. After the enzyme-substrate complex has been initiated, the chemical reaction takes place, and after we make our end chemical or product, the product will leave the active site. So if we look at an enzyme, we'll have substrates go into the active site, the chemical reaction occurs, and then products go out of the active site. And after the product leaves the active site of the enzyme, that enzyme is then capable to go and catalyze another chemical reaction. It can go bind to another substrate. So here we have two different substrates with two different shapes. Notice how only one of the substrates will fit into the active site of the enzyme. The inappropriately shaped substrate does not. So we'll have a substrate plus an enzyme. The correct substrate fits into the correct active site. We have an enzyme substrate complex form. And then at the end of that chemical reaction, we'll have two separate products leaving the enzyme substrate complex. So those cofactors are going to generally be the things that help the enzymes form the correctly shaped substrate. Microorganisms are going to need vitamins and minerals, just like we as human beings need vitamins and minerals. Trace elements, or minerals, are going to be used in our microorganisms and are known as cofactors. These can include iron, copper, magnesium, manganese, cobalt, selenium, etc., etc. Here's some random trivia for you. In human beings, sperm cells need selenium to have a properly functioning flagellar tail, which is one of the reasons why if a man is struggling with a low sperm count, oftentimes he's prescribed a selenium supplement. I digress. Back onto microbiology. So these cofactors are going to help the substrate help the enzyme form the correctly shaped active site so that the enzyme can bind to the correct substrate. In other words, the cofactors help the enzyme do its job. They help bring that enzyme and active site and substrate closer together. They can participate directly in the chemical reaction, or sometimes they don't participate directly in the chemical reaction. It depends on the specific enzyme and the specific chemical reaction that's being metabolized. Our coenzymes, aka vitamins, are organic compounds that work in conjunction with the apozyme. Remember, the apozyme is the deactivated enzyme. It's an enzyme that hasn't been fully activated yet. Once we combine an apozyme with the vitamin or mineral, or we conjugate it with the vitamin or mineral, we then are going to have a holozyme, a fully activated enzyme cofactor subcomplex. These coenzymes, generally speaking, function to remove a chemical group from one substrate and add it to another substrate molecule. Oftentimes they are going to be moving around hydrogen ions or electrons. Every now and again they'll be used to shuttle carbon dioxides or amino groups, but hydrogen atoms, so protons or electrons being shuttled by a coenzyme, is very, very common. As I mentioned before, our coenzymes are generally speaking referred to as vitamins, or come directly from our vitamins. So eat your vitamins and minerals so your enzymes function properly. Our enzymes can be classified according to the characteristics that they have at the active site, the kind of chemical reaction that they have, or the kind of substrate that they work with. Usually when we name an enzyme, the prefix or the root word in the enzyme is going to talk about some characteristic of the chemical reaction that's being catalyzed. And then the ending suffix in the name of an enzyme is "ase." Based on the biochemical action, we have 6 broad categories of enzymes. So let's look at some example names of enzymes. We can have an oxoreductase. An oxoreductase is going to have something to do with oxidation and reduction, and ase is at the end, so it's an enzyme. Oxoreductases are going to move electrons from one substrate to another substrate. A good example of an oxoreductase is going to be a dehydrogenase. It's going to remove a hydrogen ion around from one compound to another compound, either oxidizing or reducing that molecule. A transferase transfers an entire functional group from one substrate to another substrate. So if we move an entire amino group, if we move an entire carboxylic acid group from one substrate to another substrate, that's a transferase. Hydrolases are going to break apart covalent bonds that hold macromolecules together. They will go through hydrolysis. They break apart these chemical bonds by adding water molecules. A lysase is going to remove groups from double bonded substrates. It is going to lyse or cleave off those groups from the double bonded substrates. An isomerase is going to take a substrate and make it flip-flop between its isomeric forms. We can go through different chiral states. It can have dramatically different chemical properties. All of the amino acids that are metabolically available for human beings are in one isomeric state, and if they are flip-flopped to the other isomeric state, they are no longer metabolically available for human beings. You can trace the source of a protein based off of the isomeric state of the amino acids or other macromolecules present in that organism or that protein source. And an isomerase is an enzyme that will switch back and forth between the different chimeric states. And then finally we have ligases. A ligase is going to catalyze or speed up the formation of a chemical bond between molecules, and it's going to require the use of an ATP. A ligase is going to require some input of energy, and it's going to cleave off 2 hydrogens and 1 oxygen. It's going to remove a water molecule from the substrate. It's not to say that there's a water molecule attached to the substrate. Usually it's going to be 1 hydrogen and an OH groups that are separately removed, combined into water, and then spit out of the active site. So if we look at our enzymes, they can have a wide variety of pathways involved. We can have some constitutive enzymes. These constitutive enzymes are going to continue working, and they never stop working, and they never change their concentration throughout the cell. These constitutive enzymes have a static concentration in the cell, and then we have regulated enzymes. Regulated enzymes change concentrations in the cell based on the amount of substrate. So here with our constitutive enzyme in this figure, we increase the concentration of the substrate, we add more substrate, and the number of the enzymes remains the same. If we have a regulated enzyme, by adding more substrate, we cause the cell to produce more of the enzyme, and then as we have less of the substrate, the cell makes fewer of those regulated enzymes. Our enzymes' functions can be influenced very strongly by the cell's intracellular environment. Changes in pH and changes in temperature, or less commonly, changes in osmotic pressure, are generally speaking going to be used to activate or deactivate enzymes. The technical term for changing the shape of an enzyme such that you render it non-functional. So the enzyme's active site has changed shape such that it can no longer bind to the substrate. If you do that, it is known as denaturation. Denaturation is changing the shape of the enzyme so that you break or disrupt the enzyme. There are very weak chemical bonds that hold the enzymes together. Recall, enzymes are proteins. Proteins have primary, secondary, tertiary, and quaternary states. And those tertiary and quaternary states have lots of hydrogen bonds that hold them together. Those weak bonds can be broken if you change the temperature, if you have a very high temperature, or if you have a very acidic or very alkaline environment, or if you have a very salty environment, if you have a very high osmotic pressure. By denaturing a protein, you prevent that substrate from attaching to the active site, and now that chemical reaction can no longer be regulated by the enzyme. Our chemical pathways, or metabolic pathways, generally speaking, are a multi-series steps of pathways, with each individual step being catalyzed by an enzyme. The product of chemical reaction A becomes the substrate in chemical reaction B, and then the product of chemical reaction B becomes the substrate of chemical reaction C. So we'll have one chemical feed go from one enzyme to the next enzyme to the next enzyme. These enzymes will make substrates that become the products that become the substrates, so on and so forth. Some of our chemical or metabolic pathways are going to have a very linear fashion to them. Some of them are going to have a cyclic fashion that can be plotted out, and some of them can be branched. No metabolic pathways stands alone. All metabolic pathways are interconnected with each other and are going to merge at many separate sites. So, here we can have the chemical A leading to chemical B, leading to chemical C, D, and then E. And then each of these steps could have an enzyme regulating it. This is a good example of a linear pathway. Glycolysis is one that we'll talk about in a moment. We could also have a cyclic pathway, the Krebs cycle, where product will feed a product in, and then it goes through a cyclic series of chemical pathways, and then products are spit out. Or we can have our branched pathways. Sometimes we'll diverge depending on the path of travel, or we'll converge depending on the path of travel. Good examples of branched pathways are both nucleic acid and amino acid synthesis. To regulate the action of our enzymes, we'll have competitive and non-competitive inhibition. Competitive inhibition is when we supply a competitor molecule that has a similar shape to the substrate. This similar shape will mimic the active site, or this will mimic the shape of the normal target molecule or normal target substrate and clogs up this active site. And if the active site is clogged up with the competitor, the actual substrate can't get in and the normal chemical reaction can no longer take place. So competitive inhibition functions by clogging up the active site. Non-competitive inhibition, on the other hand, is going to form when we have a molecule bind to a regulatory site. Most enzymes are going to have an active site, and then they're going to have another site where a regulatory molecule can bind. When this regulatory molecule binds to the regulatory site, it changes the shape of the active site. Through non-competitive inhibition, we have a molecule attached to the enzyme at some place other than the active site to change the shape of the active site. By changing the shape of the active site, we change how its function, and we change how quickly the enzyme can catalyze the specific chemical reaction. So here we have our normal substrate and our competitor, and if our competitor binds to the active site, it clogs it up, and the normal substrate can't get in. This is competitive inhibition. Or if we have non-competitive inhibition, sometimes also referred to as allosteric inhibition, if we combine that regulatory molecule to the regulatory site, the regular active site changes shape so that the substrate cannot bind. Without the regulatory molecule in the active site, or in the regulatory site, the active site keeps its native state and the regular substrate can bind to it, and the normal chemical reaction can occur. So how do we control whether or not we even make the enzyme in the first place? Well, we could first repress the enzyme's production. If we deactivate the enzyme at the genetic level, we make it so that we stop making that specific protein molecule entirely. Now, this enzyme repression pathway, since we have to turn on and off genes for this process, generally speaking, is going to be a slightly longer metabolic pathway, or way to regulate the metabolic pathway. We also have enzyme induction. Instead of turning the genes that make the enzyme on and off, we are instead going to have our enzymes only be produced when a suitable substrate is produced or present. This is going to be the opposite of enzyme repression. This is also going to take quite a bit of time. So for our enzyme repression and enzyme induction, we can have, here we go, our DNA being bound by some repressor molecules when we have too much of the product. So our product from the chemical reaction deactivates the genes. But let's say we have a low concentration of product. If we have a low concentration of that chemical that we want in our cell, that chemical can no longer deactivate the gene. The gene will be activated. DNA gets turned into RNA. RNA gets turned into a protein. The protein forms its native shape. It forms the enzyme, and then the enzyme starts to catalyze the chemical reaction by turning the substrate into product. But as the concentration of product goes back up again, and we have more and more product, that product will then deactivate or repress the gene that's responsible for making the enzyme. So that's how we can have repression and induction. Repression occurs when we have high concentrations of the product from the metabolic pathway, and induction occurs when we have low concentrations of the product from the metabolic pathway. E. coli is a well understood bacterium that's been very thoroughly studied. If we look at enzyme induction in bacterium, we can find that if we put the E. coli in a medium that doesn't contain any glucose, but it does contain a lot of lactose, all of a sudden E. coli will start producing enzymes that will take the lactose and break it down into simpler sugars. All of a sudden the E. coli will make the enzyme lactase, an enzyme that breaks down lactose into glucose and galactose. If we take that E. coli and then subsequently put it into some medium that only contains sucrose, so a different sugar, it stops making the lactase and starts to make a new enzyme, sucrase. We're inducting or inducing the production of new enzymes based on exposing these microorganisms to new substrates. This ability to turn on and off genes as the microorganism is exposed to different food sources allows that microorganism to utilize a wide variety of nutrients, to live in a wide variety of environments, and to minimize the amount of energy it wastes by making enzymes that would catalyze or work on substrates that are not present. So, let's pause for a moment and have a concept check. Which of the following mechanisms of enzyme control breaks a reaction, blocks a reaction that's been catalyzed by an enzyme by the binding of a product to the regulatory site? So the key term here is by binding to the regulatory site of the enzyme. Is that enzyme repression, competitive inhibition, enzyme inhibition, non-competitive inhibition, or E? None of these choices is correct. Pause the video and flip back in your PowerPoint, check your textbook, or rewind the video to find the answer. The correct answer is non-competitive inhibition, or allosteric inhibition, for those of you who spend more time studying this topic in detail. So D is the correct answer. If you have any questions about enzymes or metabolic pathways, post them on the discussion board or shoot me an email. Happy studies!